Hello everyone, this is Seher from Easy Peasy, and the topic that we are going to discuss today is called as the regulation of the cell cycle. On this cell cycle, we have certain different points where the regulation will be done before entering into the next subcategory. These points are called as restriction points. They can also be called as checkpoints. So the checkpoints that are usually present on the cell cycle are M1/G1 checkpoint, G1 checkpoint, G1/S checkpoint, intra-S checkpoint, and G2/M checkpoint. There might be more checkpoints on this cell cycle, but these are the most common or most prominent checkpoints that are discovered by the scientists. Okay, now this regulation will be done by an enzyme, and that is the Cyclin-Dependent Kinases or CDK. When you hear the word kinases, it means this is an enzyme that is going to attach a phosphate group on its substrate. And it is cyclin dependent. It means that there will be a protein named as cyclin that is going to attach itself with this enzyme in order to activate this enzyme. Okay, now let's discuss this cyclin-dependent kinases in detail. So this is our enzyme, that is the cyclin-dependent kinase. You can see the structure of this kinase like this. So there is a dip over here, okay? Now this is cyclin dependent, so a cyclin protein will appear that is going to attach itself with it like this. And now, as you can see, that the structure of the CDK is changed, making a depression here, and this depression is basically the active side of this enzyme. So now this enzyme is activated, and it is ready to attach itself with its substrate. So when a substrate is going to attach itself with this enzyme, it needs the energy in the form of ATP. So they are going to attach themselves with the enzyme like this. And as we discussed before, that it is a kinase, so it is going to attach a phosphate group with it. And where this phosphate group will come? This phosphate group will come from this ADP molecule. So ATP converts itself into ADP, attaching a phosphate group to this substrate, and then they can go away from this enzyme like this. Okay, so this is the general idea how the regulation of each checkpoint is going to be done. Now let's discuss specifically with one restriction point and see how they are going to regulate that checkpoint. So the checkpoint that we are going to discuss right now is the G1/S checkpoint. Now, for example, the cell is sick and it cannot go into the next subcategory of the cell. So at this point, there will be a protein named as retinoblastoma protein that will get activated. Now this retinoblastoma protein is going to halt the process at this step and will not let it go to the next subcategory. How it's going to do it? Because it's going to attach itself with E2F protein that is attached with the DNA. For example, the cell repaired itself and now the cell is ready to go in the next step. So at this point, what happened is that the cyclin protein will get generated, that is the G1S cyclin protein. And this protein will attach itself with the CDK enzyme again like this. And this retinoblastoma with ATP is going to attach itself with CDK enzyme. And now it is going to attach a phosphate group to this retinoblastoma. And this retinoblastoma that is phosphorylated is not able to attach itself back to E2F protein. So that's how the process of cell cycle will go from G1/S phase back to S phase. And once it passes through this stage, the G1S cyclin protein is going to get degrade itself like this. So for MG1 checkpoint, we have cyclin D protein attached with CDK4 or CDK6. G1S checkpoint will get regulated by cyclin E protein with CDK2. Intra-S checkpoint is going to get regulated by cyclin A protein with CDK2. And last but not the least, the G2M checkpoint is going to get regulated by cyclin B protein with CDK1. So that's how the cell is going to get regulated on each and every step. Even at the time of metaphase, when this CE and PU protein is going to get degraded, the cyclin-dependent kinase is going to work itself over there too. So as we discussed before, that in anaphase, we have an enzyme called as separase that is going to degrade the CE and PU bound cohesion protein. This enzyme will get activated when we have the M-cyclin protein attaches with CDK and the CDK attaches with APC. APC is basically anaphase promoting complex. And as a result, this separase is going to degrade the CE and PU protein like this. And now the chromatids are free to go towards their pole accordingly. That's it for now. Thank you very much for watching this video. If you like it, please subscribe this channel. Thank you. Bye for now.